I'm greeting you all, my friends. My name is Alex Surnov, and I am that biologist who published two lectures on the coronavirus infection a month ago. Today is April 24th of the year 2020. The situation with the coronavirus pandemic has changed and has progressed pretty dramatically since the time when I published my first lecture and the second lecture too. And while sitting at home, I kept searching for the information to share with you. I hope my knowledge and my comments will be useful. If not useful, then probably at least interesting. And please welcome to the third coronavirus public talk on my YouTube channel. Before starting, I'm gonna tell you my usual disclaimer saying that my background is biological, not medical. I don't have a medical degree. This is why my words cannot be interpreted as consultation with a physician. However, I am a PhD in biology, and my professional experience allows me to analyze, systematize, and interpret the publicly available scientific publications, scientific articles that appear from time to time. You know what I mean. So I can work as an interpreter, as a translator from the science language to general audience. So right now, I think hello, good. Right now, I think we will transit to the presentation, and the remaining parts of the talk I will be conducting with the slides, with the schemes and the text. Once again, enjoy the show. So here is the name of my talk. The coronavirus SARS-CoV-2 and the COVID-19, which is the disease that this virus causes. Here is my email address. If you have questions or comments, please feel free to send me an email. And let's study first what happened since my previous lectures were recorded. How did the situation change? Well, first of all, the coronavirus outbreak definitely has transited from being just a medical and biological problem to a state of an economical problem and political and a social problem. And while thinking on the current status of this pandemic, I somehow disentangled the whole set of questions into 7 key points, just to make it easier for myself to analyze the situation. Here are the key points that I'm talking about. First of all, it is very crucial to understand how this COVID-19 disease is really going in the patient's body. What is the consequences of the infection? What the patients die of? How does the virus damage the tissues and the body? Now, the second thing is, once we learned about the pathogenesis of this disease, how to treat it urgently, how to treat it using the tools that are currently available. And if there are no effective tools that are currently available, then how to design new methods for treatment. Now, it's critically important to forecast the evolution of this pandemic. And for this, we need a lot of statistical data, and we need a good model, a model that would predict how the number of infected people changes in time, how the different strategies of therapy and prevention will affect the growth rate of the infected population, and so on. Now, it's also very important to know whom and how and when to test for COVID-19, and whether there must be a test for the virus content in the body, or it must be a test for the signs of immunity against this virus. This questions 6 and 7 are mostly organizational. The question 6 specifies how to administrate the healthcare system, meaning how to supply it with the elements of the therapy, with the necessary personal protective equipment, how to organize the transportation of the infected patients, things like that. And the question 7 is rather economical. We know that the quarantine is a valid measure against the COVID-19 outbreak. We know that it slows down the spreading, but how to introduce this quarantine in the most efficient and yet the mildest way, in the way that is not cruel for the people, in the way when we take care of the economical drop, because remember, the businesses and individual people will likely stop their activity pretty severely during the quarantine. So we need to answer how to support their welfare and how not to allow them to go bankrupt. And of course, we have to answer how to enforce and control for the maintenance of the quarantine, how to make sure that people really follow the quarantine rules. By saying we here, I mean the governments, and I'm sure the governmental structures already specified the same 7 questions or even more questions for themselves and are working as hard as they can for answering them. But I think it's worth really starting my lecture with familiarizing you, my audience, with the questions. So my professional expertise allows me to specifically talk about the questions number 1, 2, 3, and 4. I don't have good enough background to discuss the remaining 3 questions, 5, 6, and 7, and on them I can only rely on the opinion of the experts, economists, and managers of the healthcare system, and probably I have to rely on my own common sense, but you have common sense as well, and I don't think I'm a good commenter on it. I don't think I can really judge and advise you anything. So, all right, let's start with let's go through the questions 1, 2, 3, and 4, and we'll start with studying the pathogenesis of COVID-19. What do the people really die of when they have a COVID-19 when they have the COVID-19 infection going on? From listening to the physicians who treat the COVID-19 infection right now, I got the idea that the death could be associated not only with the damage that the virus directly introduces to the lung tissue, but also some other pretty damaging conditions can originate as the side effects of this disease. For example, thrombosis is the malfunctioning of blood clotting. The blood coagulates more to the higher extent in the case of thrombosis, and some vessels get clogged. So indeed, there is a report of a medical case when a certain patient had increased degree of thrombosis in the tissues during the COVID-19 infection. And also a big committee of the healthcare experts from China, specifically assembled by the government, recently issued their recommendations, and the parts of these recommendations also specifically say that the thrombosis is a likely outcome of the COVID-19 infection, and the healthcare providers and the physicians should really pay attention to the chance that the patients will develop this condition. So these links, by the way, are short here in the text. However, later in the last slides, they will be expanded, and you could see the exact full bibliographic information, about all the sources that I used. All right, that was all about thrombosis. I really don't have to say anything more than that, but I do have something more to say about the heart failure. Indeed, there is a review that summarized many previously published papers, and they state that the myocardial injury is pretty prevalent among the patients with COVID-19. Roughly 60% of such patients have some problems with the heart. And the myocards, I will remind you, the myocards is the layer in the heart that exactly generates the oscillations. It's the muscular layer of the heart that beats. So it's a very crucial tissue. And since we know the statistics, we can ask ourselves a few questions. First of all, does the virus really infect the heart itself? Does the virus fuse to the cells of the myocards and multiply inside them and cause the direct damage to the heart? The second question is whether the heart injury can be generated due to the patient's own immune reaction. The immune system tries to clear the virus. It sometimes uses pretty violent mechanisms for fighting against the infection, and as in every battle, the side effect damage is pretty likely. If you conduct a battle in a city, you likely will hit a certain building with a missile, or you will destroy a nice landscape in a park by intersecting it with trenches and so on. So the battle really requires some damage. It's inevitable. And also the third question is whether the heart injury can be the result of a very particular process that is associated with the virus fusion to the host cells. I will remind you that the virus SARS-CoV-2 uses a certain protein that is exposed on the surface of our cells to intrude into the cell. The cells are coated with multiple different protein molecules. But one protein, namely ACE2, is used by the virus to hook on it and to initiate the process of getting into the cell. And when the virus gets into the cell and starts the infection, this virus somehow downregulates the prevalence of ACE2 on the cells. So not only the cell itself gets somehow damaged, but also the number of the protein that the virus uses for fusing and for linking to the host cell goes down. And this may be damaging too, at least potentially, hypothetically. So let's go through these questions one by one. can the virus SARS-CoV-2 infect the heart? Well, the cardiomyocytes, meaning the cells of the myocardium, indeed express ACE2, and this is bad news, because then potentially, like, we can imagine that this virus will infect these cells. And also another bad news, another set of bad news is that the virus SARS-CoV number 1 indeed can infect the myocardium, indeed can infect the cardiomyocytes. The virus SARS-CoV number 1, as I mentioned already in my previous lectures, it was the virus that caused the SARS outbreak in 2002 and 2003. So we know that this virus can be found in cardiomyocytes. We probably didn't track directly the events of fusing of the viral particles with the cells of the heart, but we could detect the virus prevalence inside those cells, meaning essentially that this virus had to get in somehow. By saying we, I mean the humanity, the researchers, researchers that are cited in this review. However, some good news there are that yet SARS-CoV number 2, the current virus, has not been found in the cardiomyocytes, although it indeed has been found in the heart, but in the other cell type. There was a case when a patient had some inflammation going on in the heart, and some cells of the immune system, namely the macrophages, populated the heart due to this inflammation, and these cells did contain the virus. So how could it happen? Well, I don't know if the macrophages really got infected inside the heart, or they came already after they got infected somewhere else. They came already being infected. Macrophages are pretty mobile cells. They roam the body, and they can be attracted to the site of inflammation to enhance and provoke this inflammation. So by now we don't have the evidence that SARS-CoV-2 can infect the heart tissue by itself directly, but the news come very fast and the situation may change. We definitely will keep an eye on it. So the next question is about the heart failure. About the protein ACE. As I mentioned, the virus downregulates ACE after fusing to the host cell, and this event by itself can be pretty dangerous. Why? Well, because the protein ACE2, it's a part of our hormonal system, and it takes the hormone named angiotensin II and converts it into something else. And there are many evidence of this kind of work of this protein, and there is many details also published in these papers. Feel free to go deeper in them. For us right now, it's important to realize that you don't really want to have the excess of angiotensin II. You really want angiotensin II to be converted to its products as quickly as possible, because angiotensin II is a pretty aggressive compound. It's a vasoconstrictor, meaning that it provokes the blood vessels to shrink, and it stimulates the sympathetic nervous system. Essentially, this means that angiotensin II is the stress stimulus. It tunes the body for some kind of stress reactions. And it's a bad idea to have the excessively high level of angiotensin II on the background of ongoing COVID-19 infection. In this case, indeed, if the heart is weak, if the heart has some inherent conditions, then the heart may fail eventually. Besides that, angiotensin II was shown to be damaging in higher amounts to, the higher amounts of angiotensin II has been shown to be damaging for the lung tissue itself. The exact mechanism was not specified in the papers that I read, but what is known is that this damage is mediated through binding of angiotensin II to another protein, to the receptor for this hormone, to AT1. But this effect, I think I mentioned already in my previous lectures. So in summary, this slide tells that it's indeed something that you really don't want to have, that the excess of angiotensin II is not something you want to have during the COVID-19 infection, and it may contribute to the damage that this infection provides to the body. All right. Now, let's talk about the immune response. Can the immune response of our own body be damaging to it on the background of COVID-19 disease? Well, the immune system really monitors the body constantly and continuously for potentially foreign molecules that it can induce the immune response against. And usually when the immune response is there, it's worth initiating this immune response against pathogens, because the damage that the immune response can induce by itself is much smaller and much easier to recover from as compared to the damage that the potential pathogens could induce to the body. However, in some cases, the condition named as cytokine storm can also appear, and this condition essentially is the ejection of extremely high concentrations of the cytokines that provoke the inflammation. It is the excessively strong immune response. So what the cytokines are these little proteins that the cells of the immune system use to communicate between each other. As you can imagine, the cells like macrophages or lymphocytes or whatever, they don't have cell phones, they don't have email. All what they have is some soluble chemicals that they can emit and that the other cells can receive. And by exchanging these chemicals, they either organize their work to flock together or instead tell each other to scatter around and keep searching, or maybe these chemicals can provide the activation of these cells, or vice versa, inhibition. It is a very elegant and a very fine-tuned system that usually in most of the cases works in a balanced manner and fine, but sometimes after a severe outbreak of the infection, these cytokines can go crazy and the cells may start emitting them in excessively high amounts. So here is the paper that pulls previously recorded data from some patients with COVID-19, and they had some patients who were discharged from the hospital, and also some people who unfortunately died during the infection. And these authors measured the concentration of the cytokine named interleukin-6, IL-6, in the blood of these patients. Um, I must say that very little papers really study all cytokines at once. Most of them focus on just a few of them, on several cytokines. And this is why it's relatively hard to get the composed idea about all components of the cytokine storm, how the cytokine storm really goes, and also remember that the statistics is being completed yet. The number of cases grows, right, unfortunately. So we may not have yet all the data necessary for a good statistical overview, but for some cytokines we have individual data. And here these authors report that in the patients who were discharged, the 25 to 75 percentile interval for the level of interleukin 6 is from 5 to 9 picograms per ml. What does it mean? Well, essentially it means that a quarter of these discharged patients during their infection had the level of interleukin 6 lower than 5. Another quarter had this level higher than 9, and the remaining half of the population had the level of interleukin 6 in the ranges between 5 and 9. But the peculiar fact is that the people who unfortunately died during the infection had this interval being shifted towards higher values. Their 25 to 75 percentile interval takes the values from 8 to 16 picograms per ml. As compared to, for example, the normal level of interleukin 6, which barely exceeds 4 picogram per ml. Here I mention not the 25 to 75 percentile interval, but rather, I guess, about 90% of probability that this level of interleukin 6 will fall in this interval. So really, very little healthy people have interleukin 6 higher than this, according to this paper. By the way, thanks to a person named Ilya Sirlov, who actually pointed this paper to me in some kind of social network for scientists where you can ask for the expertise of more experienced colleagues and ask some advice or give some advice as well. Okay, so the cytokine storm is there. We can hypothesize that the level of interleukin 6 correlates to, or we can conclude that the level of interleukin 6 correlates to the severity of the disease, and we can hypothesize that interleukin 6 is a potentially damaging agent when it is emitted by cells in larger amounts. And maybe as a potential therapy, we could think of somehow blocking or inhibiting, at least partially inhibiting interleukin 6 to avoid these crazily high levels in the patients. Maybe that would help. So additional studies are needed for this, and such a therapy is already being tested. So what's next? The immune response is not constrained by the interleukin 6 emission only. Of course, there are some other interesting data about the immune response during COVID-19. This is a link for a very good overview. It is broad and at the same time pretty profound. It has much of data about many different aspects of COVID-19, immune response against COVID-19 and the infection itself. And what is mentioned there is that, for example, some people seem to have genetic predisposition, or at least, if not predisposition, then at least genetically prone to be more susceptible to the first SARS virus, the SARS-CoV number 1, that caused the 2002-2003 pandemic. So what these HLA variants are, in brief, they are certain genes which are very important for the immune system. Those genes encode proteins that hold the potentially foreign molecules and demonstrates those molecules to the T cells. So once again, the T cells, they're the key players in the initiation of the immune response, but they cannot really react on the molecules that are flowed around them. In order for the T cell to make a decision whether a certain molecule deserves an immune response against it or it doesn't, this molecule has to be demonstrated to this T cell in a particular manner. And the device that demonstrates these molecules to T cells is exactly the protein that is encoded by HLA genes, or multiple proteins that are encoded by HLA genes. Those proteins are expressed on many cells. Some cells are professional antigen-presenting cells, and they can capture the molecules from outside, from the environment, from the extracellular like fluids from the tissues, and demonstrates those molecules to T cells. Most of other cells cannot do that, but instead they demonstrate the molecules that those cells make by themselves. They demonstrate those molecules to the T cells, and they also use HLA for that. And the point is that if a cell is infected with the virus, then this cell will necessarily produce virus proteins as well, viral proteins. And those proteins will be eventually demonstrated by that cell on its surface, on the cell surface in the complex with HLA, and the T cells can come by and see this viral protein and initiate the immune response. That's how it works. Essentially, what it means, what this first row means, is that some people, the people who have certain variants of these proteins, fail to demonstrate the viral proteins to T cells efficiently enough. And the T cells just, or I mean, maybe this molecular forceps, this little device that shows the foreign proteins to T cells, maybe it's somehow loose, or maybe it doesn't hold the specifically proteins from SARS-CoV number 1 fine enough, and then the T cells just cannot react to this protein. Okay, also, let's move on. Also, this review publishes some more data on the components of the cytokine storm. In addition to interleukin-6, there are some other cytokines, and some data provided pretty similar to the data that were on the previous slide. I'm not going to stop on it. What is interesting also is that there is some necessity to the reviews like this, they mention also necessity to test for the antibodies that the patients produce against the virus. So what the antibodies are, they are soluble proteins also that bind to foreign molecules pretty much in the same manner as the T cells react to foreign molecules. But in contrast with T cells, antibodies can bind to foreign stuff that floats around. The foreign proteins do not have to be specifically shown to the antibodies. The antibodies just find them by themselves. And during the course of the disease, the antibodies change. When the infection just starts, the body of the patient, the patient's immune system produces the antibody of the class M. And here they are described as, not described, denoted by this combination of letters IgM. Ig is the immunoglobulin, it's just a fancy way to say an antibody. And IgM stands for immunoglobulin of the M class. And when the disease progresses and the immune response gets more and more advanced, the immune system starts producing the antibody of the class G instead of M. And this time kinetics demonstrates that it may be useful to test for both of these antibodies. Like, for example, if a certain person, if you tested them for the virus content in the body, and there is the virus, so the person is infected, and this person has, and you tested him or her for IgM and find out that they're IgM positive, but IgG negative, then that outcome would mean that this person just recently got infected, and he or she yet may not exhibit any symptoms of this disease, and may not even feel sick, but already perfectly may be contagious, and also in some short time perfectly may require some sort of medical help. So it's important to control for people who are the virus positive and IgM positive too, but IgG negative. In contrast, there is another situation when a person may be tested for the virus and come out virus negative, and IgM negative too, but IgG positive. What does this mean? Well, this may imply that the person has already recovered. He or she was infected previously, successfully cleared this infection, and now is immune. The IgG is there in the body. And then such a person can be let to walk around because neither he or she can be infected secondarily, nor they can be contagious anymore. So that's a possible strategy of controlling whom for whom to establish the quarantine and who are safe to leave the quarantine. And also, this IgG appear to be a potent agent for the therapy, because we can drain the blood from this recovered person. We can take his or her IgG and transfuse it to the people who are suffering from the infection currently now, to the people who are short in their own IgG, right? This therapy strategy is being discussed too very extensively and even applied somewhere in some regions. All right, so also this review has some links and references to the data on perspective treatments, perspective therapies, and let's proceed with discussing the therapies right now. So first, the currently available treatments. Unfortunately, here the situation is not has not that dramatically changed since the last lectures. Still, the therapies that are very well established and by 100% recommended are not specific to the virus. They essentially just make it easier for the patients themselves to go through this infection, meaning that if this person experiences trouble with breathing, if the lungs are deteriorated, well then please pump the oxygen in these lungs. if this person lost a lot of water from the body due to the immune response and the infection, then supply this patient with the water and electrolytes. If there is a secondary opportunistic bacterial infection going on in the lungs because the body cannot really cannot suppress proliferation of this bacteria anymore on the backgrounds of COVID-19 infection, then yes, treat please this bacterial infection with the antibiotics. However, none of these approaches really targets the virus specifically, and they do not target the damage that the virus introduces to the body by itself. However, a lot of researches are going on, and a lot of data is being generated, and some perspective treatments also are being extensively discussed. And hopefully, in the closer future, we will have at least some tools to target the virus specifically. For example, in my previous lectures, I already mentioned the combination of these two antiviral drugs traded under the name Caletra, and I said that they were quite effective against the previous SARS pandemic, against SARS-CoV-1. Unfortunately, during the current pandemic, the studies did not confirm that this combination is effective. It may be really a true result, and we're just having bad luck with these two virus drugs. Alternatively, the statistics, the number of patients, the cohorts of the patients could be small and insufficient to really capture some minor effects of these drugs. But also, I want to mention here that it's pretty hard to conduct the trials of certain drugs or therapies or approaches that would by 100% satisfy all formal requirements for a good medical research. Not because the authors are bad, not because the research is kind of somehow inaccurate, but just because we're going out of time, we're playing against the time. The infection is getting spread very rapidly, and some demands of this medical rules can be just inapplicable in the current situation. For example, this study was randomized, meaning that the people were allocated to experimental and control group by chance, randomly, but they were not blinded, meaning that the doctors knew which drug, whether the current patient was given with a drug or was left without a drug. And in most of the cases, it's probably fine. It doesn't really affect the medical outcome. But we can imagine the situation when in a study like this, I'm not talking about this exact study, but we can imagine really a situation in a study like that when the awareness of the doctors about the actual strategy of the experiment would lead to some subconscious bias. The doctors even may not deliberately, not intentionally, but they can go a little bit biased with assessing and estimating the improve of the state of these patients, and when they judge whether the patient got better or not, they may subconsciously use the knowledge of the strategy of the treatment. This is why really the perfectly good experiments should be conducted in such a way that the person who assesses the states of the patients and the person who really records the medical history of the patient should not know what exactly the patient is given. What the patient is given should be known only by the administrator of the experiment. Now, another potential drug that seems effective previously is favilavir, and indeed there is some evidence that this drug is effective, but in this case the study was not only not randomized, but even there was no really control group. The authors somehow compared to some other data that were available. And again, the situation may change, new research may appear, we still can find some more promising data on favilavir and on these two guys, but right now I guess we just have to keep our minds open and keep looking for newly appearing data. By the way, the article that studied the effects of favilavir is indicated as being retracted from the journal. I didn't see it yet on the journal's webpage, but the software that manages my bibliography, the Zotero software, came out with the notification that this paper is now retracted. Okay, anyways, keep our eyes open and look for more appearing data for the data that for the data that appear anew. All right, go on. Here I have a link for a review that summarizes some perspective treatments. Sorry, I need a sip of water. So how to target the virus specifically? This virus has a few components that are principally absent from our own body. For example, our genetic information is recorded on DNA, but the genetic information of the SARS-CoV-2 virus is recorded on RNA instead. And this is why for its replication, for its proliferation, this virus should use a completely different molecular machinery from the machinery that our own cells use. And thus, this machinery can be a pretty appealing target for perspective treatment, because by targeting this virus replication machinery, we almost for sure will not damage our own molecular systems, because we simply don't have anything similar to that. And indeed, there are two drugs being right now developed, being under the development process right now, which targets the RNA-dependent RNA polymerase of the SARS-CoV-2 virus. Also, the virus does need to cleave some proteins during its life cycle. Some proteins need to be cut in parts by specific molecular scissors that are called protease. So some approaches targets this viral protease as well, and also two drugs are mentioned here. Also, the virus cannot multiply and replicate itself being outside of the host cell. Outside of the host cell, every virus is just it's lifeless. It's not life. It's just a crystal structure of nucleic acids, proteins, and probably lipids, but it cannot grow. It cannot do. It manifests the signs of life only when it intrudes into the host cell, and and this is why if we effectively inhibit the fusion of the virus particles to cells, we're safe. We, in this case, will not have an infection whatsoever. And indeed, some drugs are being studied that can block potentially this virus to cell fusion. Well, one of these drugs I wrote here that it doesn't work. In fact, it's not true. It does work, but it works significantly worse as compared to others. Let's put it this way. Anyways, you can go into this review and see the details if necessary. Some negative examples are good too. It means that the natural selection of the drugs is also happening. All right, so what else? We can manipulate the immune system. As I said, the immune system can be very helpful. Sometimes, in rare cases, it can be damaging. So we really have to focus on the strategies of enhancing the immune response, or vice versa, on strategies of attenuating the immune response. Some drugs are mentioned here that serve these purposes too. And of course, vaccines. Vaccines are being developed, and moreover, the vaccines that are now being developed against SARS-CoV-2 are usually based on principally different approaches that the approaches that are principally different from those that underlie the vaccines that we know, the previously available vaccines. So, for example, all these vaccines that we have received, MMR, the vaccine against tuberculosis or smallpox, they are pretty old, if not to say archaic. They were designed long time ago. Probably most of them are based on attenuated or killed pathogen themselves, right? So it is either a killed bacterium or a semi-destroyed virus, or a virus that has been passed in cell culture for long enough that it lost most of its damaging ability. But right now, against SARS-CoV-2, completely different approaches are receiving some green lights from the healthcare control organs. And for example, we can genetically modify the antigen presenting cells. Those cells that demonstrate the antigens to T cells, as I mentioned already previously, well, we can insert short segments of genetic information into these cells and make them to produce and expose the proteins that are recorded in this genetic code. And this can be a viral protein. We take a short nucleic acid, we record a structure of the viral protein on this nucleic acid, like we record the files on a hard drive or on a flash drive, whatever, and we insert this nucleic acid into an antigen presenting cell. And then this cell will produce this viral protein by itself and no other components of the virus particle. So the infection principally cannot develop in this system, but the antigen presenting cell will have something to show to T cells, and the T cells will get trained after seeing this virus, single virus protein exposed on the surface of antigen presenting cells. That's a good approach, pretty promising. A similar approach is to take this virus RNA and just modify it in such a way that it cannot replicate anymore, and just put into the body, encapsulate it in a certain virus-like particle as well. So this particle will for sure, principally, be unable to proliferate, but it will be able to go inside cells and to translate its genetic information into virus proteins. And then those proteins will get shown to T cells. So those two approaches, and by the way, this second approach seems to have the best shot. It seems that exactly this, if nothing really bad happens, then this vaccine will be approved soon. That's my guess, and that's what the experts are saying. Going on, here is the big table taken from the review written by Persian authors from Iran, and they just summarize different therapeutic strategies that manipulate the immune system somehow, so-called immunotherapy. And for those of you who are experts or just interested in this, feel free, please, to download this paper and go carefully through this table, and feel free to go carefully through these links. These links are inherent to this paper, right? When you go in the paper, then those references will be in it. These numbers, you know what I mean. These numbers mean the intrinsic reference list within the paper. But nonetheless, many coronaviruses, according to this table, many immunotherapy approaches target different coronaviruses, according to this table. So please feel free to look at that. Now what? I guess we're transiting to the modeling now. The models are important to predict the developments of this disease in the future. For having a good model, we also need to have a good statistical data. Here is the web page, the full address of which will be available in the end, and it has a lot of interactive charts with different statistical information about the COVID-19. Such a huge abundance of statistical information makes me to believe that most of the governments, at least in the developed countries, do supply the healthcare organizations with actual and up-to-date information. They really do a good job in measuring how many people are infected, how many people have recovered, how well the quarantine works, and this information gets accumulated and used in the models. However, I perfectly can imagine the situation when some countries fail to provide such an up-to-date information. Why? Well, maybe deliberately, due to censorship or whatever, maybe because the testing, the tests are not that available in these countries, or maybe because the statistics is not really well developed. since we can suspect at least some countries to be not that adequate in providing the information, it is probably safer to develop only geographically local forecasts. Of course, ideally, it would be the best to have the forecast for the entire world, but in the current situations, maybe our best shot is the geographically local models. And indeed, since the quarantine pretty much suppressed by almost 100% the passenger transportation across the world, at least intercontinental transportation, we can hope that the progress of this disease is dependent primarily on the situation within this country, but not on the external perturbations. CDC on their webpage refers to 3 different models, and those models use absolutely different approaches. However, their results converge to a pretty similar picture, and that's nice. That's nice when something very different in the end shows more or less similar results. It increases the confidence of these results. So let's go through these models one by one. The first model just takes the experimentally available measurements. And by the way, this is not an actual graph. This is just a scheme, what they do. So this model takes available statistical data, like the progress of death rate in time, and they fit those data with a searching formula. They took first this kind of formula and then say, no, it's not a fancy enough formula. We rather will fit this data with this formula. Why? Well, just because this formula fits them better. There is no mechanistic insight behind choosing this formula over this one. Just this one works better, period. And well, then you can ask, all right, fine, why don't we just connect those dots with straight lines and skip using any formula? Well, the key point here is that by reducing all this data, all this statistical data to just one little row, we can, first of all, simplify the calculations rapidly, but secondly, we can forecast to the future. We can predict or try to predict how this curve will grow in time. So, for example, we take all these red dots, we choose the blue line that is described by this formula in such a way that it fits the points the best, and then we see which parameters this line corresponds to. You see this function depends on the parameters alpha, beta, and p. So we choose such parameters that fits the red dots the best. And then we just insert here the time from the future, let's say the time on the next week. And we, by doing this, we calculate the approximate progress of the death rate within the next week. Well, it always may change, some new data may appear, and the fitting may require correction, but for some short periods of time, this model does provide a good forecast. The next model, or the next forecast algorithm, was not really specified exactly. I couldn't find the exact article that says what they really are doing, but as I understood, they do not use any exact formula like here. Instead, they estimate what is the chance for the death rate or any other parameter on the next step to leap to a certain value from the previous one. They measure the distance between this dot and this dot, and they measure the distance between the third dot and the second one. They measure all these little increments, compare them all to each other, and then they generate a prediction. What is the chance for the next dot to make a leap with the magnitude, with the given magnitude? What is the chance for this next dot to jump here, and what is the chance for it to jump here or here? And then they kind of and then they calculate that the chance that this dot will jump here to this point is pretty low, and I painted this region in light blue. The chance that it will jump here is a little bit higher, and they painted it darker. Here there is the most of the chance, and so on. Right? So by comparing those jumps, those leaps, the magnitude of these leaps to each other, the authors somehow calculate the probability of the further progress of this disease. And the third model used indeed models mechanistically all the underlying processes. I didn't draw graphs here, I rather drew a computer where the authors wrote equations, and those equations exactly account for the chance to meet the virus, the chance that the virus gets transmitted between different people, the chance that the people will travel in the world and the direction that they will travel to, and all these exact physical processes are captured within the equations. It's a very complex set of equations, of course. It operates on multiple different levels, and the level of virus and the transmission of this virus between people, and also some local travels and the global travels. You see this globe here. I put those arches which depict the direction of the air flights, how the plane fly. So in one word, this model takes into account many different processes, takes into account their probabilities, and come out with the predicted kinetics of this disease development in case when there is no quarantine, in the case when the disease is we try to suppress this disease somehow, in case of current treatments, in case of potential treatments, and just in many different cases of our actions, what we can do. Pretty much the same as we forecast the weather, honestly, right? We also measure the airflow, we measure the pressure, barometric pressure, we measure the degree of precipitation, we know the mechanistic laws of physics that govern motion of air masses, and by applying them and by running them through the time, we forecast somehow what the weather will be. All right, I guess that's mostly it. I have to say a little bit about testing as well. There is an article in The Economist, and this is the first article in The Economist actually that I read, and the authors there compared the outcome of the testing for COVID-19 among the population in Iceland and the population in the Netherlands. So in Iceland, they tested people randomly, not looking at whether they are suspected for having COVID-19 disease or not, just the entire population. Or at least they sampled people randomly from the population. And in the Netherlands, they tested only the people who got in the hospital and who are suspected for having COVID-19. And in the Netherlands, it appeared to be that most of the SARS-CoV-2 positive people are of the older age, whereas in Iceland the population was the positive population was younger. So one of possible explanations of this fact is that the young people get infected with the same rate as the old ones, but they are not that likely to develop the disease itself. So the chance that a young person after being infected with COVID-19 will stay asymptomatic is higher than the same chance for the older person. Maybe some other explanations are possible too, but nonetheless, I guess it introduces it points out that there is a risk that a lot of young people who are currently now feeling healthy and just walk on streets are indeed asymptomatic carriers of this virus and may be contagious, unfortunately. That's what I wanted to say. As I mentioned previously, I cannot say anything about the economical aspects of the COVID-19 diseases, so you better just listen to the experts in these fields. I can show now the references, and you're welcome to press the pause button and look at all of them precisely and go into any of them, Google for any of them. Here it is. Thank you for staying with me. I hope it was not boring. I keep my eyes open. I will make another presentation in case if I have something necessary to say. See you at some point. Have a good day.